Well, welcome to Chapter 8, and we're going to talk about cell signaling systems. And cell signaling systems are so important because this is the link between what happens in the extracellular environment of a cell and the changes in cellular behavior. So there are many different ways that this signaling can happen, but the common feature, the common thread is that the signaling systems all have to amplify the signal, and they amplify that signal through enzyme-mediated reactions. And so we're going to see a bunch of different ways how that happens. But basically you have some kind of ligand, right, that's going to bind a receptor protein. You're going to induce conformational changes in the receptor, and then you're going to affect some kind of downstream biochemical activities of our proteins inside the cell, right? So that's our kind of general outline of what happens. And so what we'll do is we'll talk first about the components of cell signaling pathways. So what are the general ideas? How do they generally work? And then we'll talk specifically about a couple of different ones. The big four that we're going to cover are G protein coupled receptors, tyrosine kinase receptors, tumor necrosis signaling, and then nuclear receptor signaling. So it's good to have kind of examples of each one of those big four so that we can understand the process. So we know that components of our signaling pathway are always going to include some kind of extracellular messaging protein, right? This is our ligand. This ligand is going to bind to the receptor protein, and that's what's going to induce a cellular response. So until that ligand binds, nothing's going to happen. So how do you trigger a cellular response? Well, just like we've seen in the past, once a ligand binds, you can induce a structural change inside of that receptor protein. That structural change is initiated by the binding. So it's really nice in biochemistry. Finally, we've got to some things where things are named correctly, right? Where the name of the receptor reflects what ligand activates it, right? So if you have, for example, glucagon, right? That is the ligand, and that ligand is going to activate the glucagon receptor. Oh, magical, right? So nice. Insulin. Insulin's going to activate, guess what? The insulin receptor. Oh, finally they named something well, right? You also have ligands that can pass right through the membrane of the cell, like, for example, estrogen, right? And then that's going to go and bind to a nuclear receptor. But it's the same thing. Estrogen is going to bind to the estrogen receptor. It's just that that estrogen receptor is in the nucleus versus on the outside of the cell, but it's still named beautifully, wonderfully. Don't you love it? All right, so we call the mechanism responsible for transmitting our extracellular signal across the plasma membrane signal transduction. So transduction, right? Transduction in general means a change, an induced change. So what's happening is you're changing from information into a chemical signal. And so usually at the end of this signal transduction pathway, you're going to end up with a covalent or sometimes a non-covalent modification of our intracellular target proteins, right? Our target proteins are what are at the bottom of the pathway. The receptor proteins are at the top, right? And the target are at the bottom. And so the target proteins are what are going to control, these are going to control our cellular responses, right? So there's like a signaling cascade that's going to happen in order to get to that target protein, and that target protein is going to control our cellular response. So what are some examples of things that can be modulated by signal transduction? Metabolic pathways. Which way you're going in the metabolic pathway can be controlled by signal transduction, and we'll look at an example of that. Ion flow through the plasma membrane. We're going to look at an example of that. Ion flow is super important. We learned about that in muscle contraction in the last chapter. Cell mobility, right? So how the cell is going to move around. Not all cells are mobile, but the ones that are controlled by signal transduction. And then also for those nuclear receptors, gene expression. That's a big one. Okay, so consequences of activating a receptor protein. You can have a couple of different things happen once you activate that receptor protein. You can have a covalent protein modification. So the ligand binds to the receptor, then that receptor is going to either phosphorylate or dephosphorylate or some combination of both of them, some protein modifications there, right? You can have protein conformational changes, right? So maybe these are non-covalent interactions between adapter proteins. So these are adapter proteins come after the receptor, but before the target protein. You can change rates of protein expression, right? We talked about this with our nuclear receptors, right? They can occur at the transcriptional level. So this is RNA synthesis and RNA turnover. And the same thing translationally, that's protein synthesis and turnover, right? So either one is possible. So we call this pathway, the cell signaling pathway. And so it's a linked set of biochemical reactions. You have a bunch of biochemical reactions that are first initiated by that ligand binding to a receptor protein. And eventually that signal is going to be terminated. Like you're going to have an endpoint. The start is the ligand induction and the end is your termination. So I'll give you an example. It's so hard to talk about this and not give you examples. So for example, insulin signaling pathway, right? You have elevated levels of, you have high levels of insulin in the blood, right? In the bloodstream. So what's going to happen? You're going to have insulin binds to the receptor, right? Once that insulin receptor is activated on the surface of our target cells, now we're going to have this series of biochemical reactions, right, that are going to occur. And it's not just one, it's usually a bunch of them, right, that are going to involve activation of intracellular protein kinases. So activation of intracellular protein kinases, right? What does a kinase do? A kinase is going to phosphorylate something, right? And ultimately, we're going to regulate glucose homeostasis. So ultimately, glucose homeostasis, right? And so you have to understand it's a very, very, very complex process. And each of the different ligands are going to bind to a different receptor and cause a different downstream response. So I should explain that. When we're talking about a signaling pathway, inside of that cell signaling pathway, you can have early steps, and those early steps are referred to as upstream events. And then late stage or later steps are our downstream events. And you'll see this is like our target protein modification. Right? So in the upstream events, we have first messengers, right? These are extracellular ligands that are going to bind to our receptor proteins. Then you're going to have the next wave is second messengers. Second messengers are intracellular only, right? So our ligands are going to be first messengers. Then inside the cell, we're going to have intracellular signaling proteins, and those are our second messengers. So first messengers are our ligands, right, that we've been talking about, and second messengers are all intracellular. Kind of remember that. So here's kind of a diagram that kind of walks you through one possible example, just one, right? So you first have your first messenger, right? This is your ligand. that's going to be received by your receptor. So this is our upstream, see how they've labeled it here, upstream signaling events. Now, we have one ligand that binds. We need to amplify that signal through activation, excuse me, of one or more downstream target proteins. So here's an example, right? We have some upstream signaling proteins. These are ones that are very close to the membrane, that are very close to the receptor protein. Then you're going to have second messengers, and these second messengers are going to induce the downstream signaling proteins, right? So for example, a downstream signaling protein might be a kinase that then goes in and phosphorylates all these target proteins. And each of these target proteins is something different. One's a transcription factor, one's a metabolic enzyme, one's a cytoskeletal protein, one's a transport protein. So it's going to target a variety of cellular proteins. It's usually not just one. It can be, but usually not. So like, you got to kind of think about this. How important are signal transduction pathways? Well, if you're a unicellular organism, it's not that big a deal, right? Because usually signaling pathways are referred to or used for, are the conditions suitable for reproduction, something like that. But for complex multicellular organisms, eukaryotes, it gets really, really important. So if you look at, for example, yeast are, if you look at their genome, right? If you look at the percent of genes that are involved in signal transduction, in all this junk that we're looking at here, right? Yeast have about 3% of their genome. I'll write it. Come on. Yeast is about 3% of their genome that's signal transduction pathway coding sequences, right? Roundworm, we get a little more complex, a roundworm. But we're still unicellular here, 6%. Now we get to humans, right? I've been a big jump there. Humans are about 9% of the genome is just signaling pathways. I mean, that's huge. That's almost 10% of all of the genes inside of our DNA that are devoted to cell signaling pathway. And I'll tell you that this number does not include transcription factors, even though I kind of think it should. It doesn't. So it's a huge, huge portion of our genome. So it's very, very important. So let's talk about some of these first messengers, these ligands, right? Hormones can be ligands, right? They're biologically active compounds that are made somewhere and then released into the circulatory system, and they go find their target cells and bind to the hormone receptors on those target cells. And so when you look at them, we categorize them in 3 different ways. They can be endocrine, paracrine, and autocrine. So if you think about this, endocrine, basically what you're going to do is one cell is going to make it. So here, over here, right? So you have this one, let's do this as endocrine. This is the cell, makes the hormone. Then that hormone is going to travel a really far distance, and it's going to get to a distant location. And so it's going to affect the activity of a distant cell. So distant cell responds to hormone, right? Then you can have things that are paracrine and autocrine, and these are local. So a local cell is going to make it. And there are 2 different ways that locally that can happen. The first one I'm going to talk about is autocrine. Autocrine means that one cell, this blue cell right here, makes a hormone. It comes out into the extracellular environment, and the same cell type picks it up. So autocrine, same cell type, makes and receives signal, right? Paracrine, one target cell, right, is going to make it, and it's going to go to a close location, but it's going to be a different cell type. It's a different tissue type, right? So it's still local. Didn't have to go far, but it's a different cell type. So local, but here's a different cell type receives signal or receives hormone, right? So you have to understand the difference between endocrine, paracrine, and autocrine. Autocrine's easy. Auto is self, right? So one cell makes it, same cell responds to it. Para, I don't know, it's close, but endocrine, it goes all over. Okay, so hormones are not the only ones, right? If you look here, for example, estradiol, this is a hormone. Insulin is a hormone. Let's see, testosterone is a hormone. So this is an example of a first messenger, but it's not the only first messenger. First messengers could be ions. First messengers can be small molecules. We're going to talk, for example, today about nitric oxide and how nitric oxide acts as a first messenger. And so in nitrous oxide, it's generated in cells by this enzyme called nitric oxide synthase. Hey, they named it correctly. Woohoo, right? Nitric oxide synthase. And it happens as nitric oxide synthase converts arginine. It goes through oxidative deamination of arginine into making citrulline. And so a product of that is nitric acid, nitrous oxide. So you have rapid diffusion of nitrous oxide into smooth muscle cells. And once smooth muscle cells receive that signal, you get muscle relaxation, you get vasodilation, you get increased blood flow. So all of this is really great, but you have to understand how nitrous oxide signaling is mediated. Right? Nitrous oxide signaling is mediated by the activation of a soluble enzyme called guanylate cyclase. All right. And so this generates our second messenger, which is called cyclic GMP, right? So right here, cyclic GMP. So cyclic GMP is really important in this pathway. And if you look at biochemical pathways, drug manufacturers try to manipulate these biochemical pathways in order to treat illnesses. So there was a drug in the 90s that was designed to treat heart disease. And it was designed to inhibit cyclic GMP phosphodiesterase, which is an enzyme that hydrolyzes or gets rid of cyclic GMP. GMP. And so if you took this drug, right, the cyclic GMP would become deactivated. And the drug ended up being not very effective in terms of heart disease, but it had a really interesting side effect. And so if you see that it's a blue pill, this blue pill was renamed Viagra, right? And it had this interesting side effect of having prolonged penile erection. And so this is because our cyclic GMP is not hydrolyzed. And so you end up having an increase in our steady state levels of our cyclic GMP. And so the cause, the nitrous oxide effect basically doesn't get diluted. And so you end up in those steady state high levels of cyclic GMP, which results in prolonged vasodilation. And so all of the drugs in this category basically work in a very, very similar function on cyclic GMP. So things you always wanted to know, right? But cyclic GMP is not our only super important secondary messenger, right? They're all small, non-protein, intracellular molecules, but they're all designed to amplify receptor-generated signals. You get a small signal in, and we want a big effect in the cell. And so you'll hear about cyclic GMP, cyclic AMP, right? So the AMP, the GMP, that's all cyclic adenosine monophosphate, right? Diacylglycerol, which you'll hear abbreviated as DAG. Inositol 1,4,5 triphosphate, we call that IP3. And then calcium is also, the calcium ion is an important messenger, right? So let's talk a little bit more about cyclic AMP, right? So here's our structure on the side. We start out with ATP and through adenylate cyclase, and we have a loss of 2 of our inorganic phosphate groups. And then we have a cyclic molecule that results. And so this is why we call it the cyclic AMP. And so cyclic AMP can then be converted to AMP, but this molecule right here has a huge effect on the cell. So let's talk about what that effect is. If we have cyclic AMP being made, the cyclic AMP is going to bind to and activate a downstream signaling protein. So we have our cyclic AMP here, right? It's going to bind to an enzyme called pKa. pKa is protein kinase A. Can you guess what it does? It's a kinase. Yeah. So pKa is going to amplify our signal even more. It is going to go in, it's going to phosphorylate a bunch of different targets. One of those targets is phosphorylase kinase. It is a kinase, it is not a phosphorylase, but it is a kinase. So that enzyme 2, phosphorylase kinase, can go in and activate a third one, glycogen phosphorylase, right? And so it activates glycogen phosphorylase. And this enzyme, so enzyme 3, is responsible for the phosphorylysis reaction that releases glucose from glycogen. So we have glycogen and we convert it to glucose. So each step is an amplification of our original signal, right? You had one adenylate cyclase enzyme that was activated, just one. Then that's going to generate, oh, I don't know, 100 cyclic AMP molecules. Those 100 cyclic AMP molecules here, right, are going to activate 50 pKa molecules. Those 50 pKa molecules are going to activate 5,000 phosphorylase kinase molecules. Those 5,000 phosphorylase kinase molecules are going to cause the release, and you don't have to memorize these numbers, something like 50 million molecules of glucose 1-phosphate from glycogen. So one small signal in the beginning is going to end up in a large signal at the end. And that's the whole point of signal transduction. Big overall concept there. So we're going to talk about another secondary messenger. Well, we're going to talk actually about 3 of them. DAG, IP3, and calcium 2+, right? So when we want to control our secondary messengers, so DAG and IP3, these are 2 secondary, I guess I could go secondary messengers, right? You want to be able to control them. And so they're controlled by the activity of a membrane-associated enzyme that's called phospholipase C, or PLC. This is going to hydrolyze our membrane phospholipids. So here is a membrane phospholipid. You should know about phospholipids from lab, right? So you have this membrane phospholipid and it becomes hydrolyzed. And when that is hydrolyzed, right, we end up with 2 products. We end up with DAG and we end up with IP3. And so the enzymatic activity of our phospholipase C is going to be regulated by the activation of a membrane receptor. So you have activation of a membrane receptor that activates phospholipase C that then creates these secondary messengers. So you're seeing the cascade happening here. So let's look at that kind of cascade and what's going on. All right, so we had PLC that was activated by our receptor activation, right? So here's PLC, it's become activated. It is going to hydrolyze our PIP2, which is our membrane phospholipid, right? And we're going to create IP3 and DAG. So Now, DAG binds to and activates PKC. This is similar. PKC is protein kinase C. It's similar to protein kinase A, right? It's just the third one I think they discovered. IP3 at the same time is going to go in and activate calcium channels that are located on the endoplasmic reticulum. When those ion channels are activated, you're going to have release of calcium into the cytosol. So this is cytosol here, into the cytosol. So once we have calcium in the cytosol, now you have all these binding proteins that are activated by calcium. One of them is calmodulin. Calmodulin is going to activate a target protein. So here's an example of our target protein to induce some kind of effect. Calcium will also further activate PKC. Then PKC can go on and phosphorylate more downstream products. So you're seeing this complex cascade that is just going to amplify the signal over and over and over again. And that's exactly what happens in a signal transduction pathway. So we're going to back it up just a second, and we're going to look at the different types or the different classes of receptor proteins, right? Our receptor proteins are the information gatekeepers of the cell. They decide what's going to happen and when, depending on whatever signal or whatever ligand is binding to them. Eukaryotes have 5 different classes of receptor proteins, and each one is going to use a unique mechanism to transduce a downstream signal. So of the 5, the remaining parts of this chapter are going to cover, each section is going to cover one of the 5. The first one is a G protein coupled receptor. So in a G protein coupled receptor, once a ligand binds, you have attached to that receptor a G protein complex, and it's basically inactive and just hanging out with the receptor. And when our ligand binds, that extra complex is going to disassociate. And once that disassociation happens, then you're going to activate some secondary messengers, right? You're going to activate adenylate cyclase. You're going to activate our phospholipase C, that's PLC, right? To get that cascade going. Another class is our receptor tyrosine kinases. These are enzymes. These are enzymes, so they actually phosphorylate tyrosine residues in target proteins. And so once those target proteins are phosphorylated, now they can go and dock or go and bind to other molecules to induce that intracellular signaling. You have tumor necrosis factor receptors. These transmit extracellular signals by the receptors themselves are kind of like all by themselves, but once they bind to their ligands, they form trimers. So the receptors actually come together, and so you have 3 receptors that kind of bundle together, and these control inflammation and apoptosis, so programmed cell death. These are really cool. You have nuclear receptors, which is not going to be covered so much in this chapter, but they modulate gene expression through either protein-DNA interactions or protein-protein interactions, right? And so that's in a different part of the book. Then the last one is ligand-gated ion channels. These are really similar to passive transport molecules that we talked about earlier, and they control the flow of sodium, potassium, calcium ions, but they control it in response to ligand binding. So sometimes they're open, sometimes they're closed, and ligand binding will control that opening and closing. So it's really nuclear receptors that are the only ones that are not membrane-bound proteins, and these are super kind of special because they bind extracellular ligands. They don't bind extracellular ligands, right? So everyone else, right? They're membrane-bound proteins. That bind extracellular, extracellular ligands. All except for nuclear receptors, right? That's a definition for everybody except nuclear receptors. So let's kind of look at the mechanism, kind of a diagram mechanism of each of these. So the first one was our G protein coupled receptors. So here's our receptor in the dark blue. So this is the receptor portion. And this is the G protein. So in the inactive state, they're all hooked together. And when your receptor binds to a ligand right here in the binding site, then it's going to induce release of this G protein complex. And so this is really a preformed inactive complex that once it's released, then it becomes activated. The second type were those receptor tyrosine kinases, right? And so this, once it binds a ligand, is actually going to add a phosphate group to some kind of a target. It's not a target protein, it's actually an adapter protein, right? An adapter, it phosphorylates an adapter protein, which then that adapter protein can go in and induce changes within the cell. Our tumor necrosis factor receptor. So, oh, I kind of know how it switches to eraser without me doing anything. But all right, so activation of this one results in that receptor trimer formation. So usually they're spread out and they're relatively close to one another, but they're not bound together. But when they bind the ligand, they trimerize and they promote association of cytosolic adapter protein complexes, right? And so we'll talk about those in detail later. And then you have steroids. You have steroids that can actually pass through the membrane, no problem. And they come in and they modulate gene expression, right? So they bind to transcription factors, something like that, and moderate that protein-protein or protein-DNA interactions, depending on whichever one we're talking about, right? All right, so what I want to do is I want to talk about ion channels. Ion channels are so cool. So here's one example. This is the nicotinic acetylcholine receptor. This is going to mediate ion transport in response to the neurotransmitter acetylcholine, right? So acetylcholine, this is going to be our ligand, right? And its function is to transmit physiological signals across a neuromuscular junction. So a muscle and a neuron, that's a neuromuscular junction. And it's in response to a neuronal stimuli. So you thought to yourself, I want to raise my hand in class. So you have this neuronal signal that is going to go, from your brain down your spinal cord, down the nerves in your arm. And so this is where the nervous system connects to the muscular system. And so once that neuron is activated, right, the one that's right next to the muscle, what's going to happen is you'll have this propagated electrical signal through the neuron. And I don't know if you've gone over that in like molecular biology or something like that, but the end result is that you have these vacuoles, and they're called synaptic vacuoles or vesicles, and they have neurotransmitters inside of them. And when they get the signal that that neuron right here is activated, what they're going to do is they're going to merge with the plasma membrane right there at the synapse. And when they merge with the plasma membrane, they empty their contents into this synaptic cleft right here. And so the molecules will flow from the neuron into the plasma membrane of the muscle cell, right? And so the acetylcholine will bind to a receptor. And this receptor is the nicotinic acetylcholine receptor complex, right? So here's the binding site for our acetylcholine. And so the ligand binding is going to induce a conformational change. And that conformational change is going to open the receptor. So you see how it's closed here and it's open here. So once that channel's open, all of the sodium and the potassium ions are allowed to flow. So we have sodium and we have potassium ions that then can flow into the cell, right? When sodium and potassium ions flow into a muscle cell, what's going to happen? You're going to have membrane depolarization. When you have membrane depolarization, do you remember what happens in a muscle cell? We talked about this. You have calcium release, release from our sarcoplasmic reticulum, from the sarcoplasmic reticulum. What happens when you have calcium release from the sarcoplasmic reticulum? Oh, now the calcium can go and bind to the tropomyosin. That's going to expose the sites on the actin, and so on and so on and so on, and you're going to get muscle contraction, right? Muscle contraction, okay? So all of these pathways are super complicated, I know, but when you start to see the commonalities between them, it starts to make them a little bit easier to understand. So we've kind of gone through the big ones, and you have a general overview of what they're like. And so in the next section, we're going to start with our G protein coupled receptor and kind of see how that one specifically works. So have a great rest of your day, and that is the end of 8.1.